Morphological and anatomical study of the supply specimen, that is Selaginella species. And Selaginella is of different distribution and they have different type of species available and distributed throughout the world. And here the aim of the experiment is to study the morphology and anatomy of the supply specimen, that is Selaginella. And we have collected Selaginella as a bottle specimen, and we can also brought it by through these different agencies. And for these spectacles we need different materials for our study, that is needle, blade, safranin, methyl green, microscope, slide, cover slip, pen, paper, plant specimen, etc. These are the specimens, and we have to take all the specimens before starting our practical. The procedure for the morphological study, we have to observe the habit of the plant. We have to note down the characteristic features of stem, root, and leaf of the plant, and also have to observe the reproductive structures seen in the specimen. For anatomical purposes, a very thin transverse section of stem and leaf are taken separately, and each of them is transferred through double staining process and observed under compound microscope to note their anatomical behavior. For the double staining process, for the staining of the sections of the root and as well as the stem, we are using safranin, and the sections after cutting are deep under the safranin and keep for 3 minutes for stain. Then the safranin is removed with the help of blotting paper, and immediately 70% of ethanol is added. That is, in 70% of ethanol, the sections are transferred. After 1 minute, the ethanol was removed with the help of filter paper, and again 70% ethanol was again added for 1 or 2 minutes for remove. Again, the 70% from the 70% ethanol, blotting sheet are used to immediate dry the ethanol from the sections, and methyl green stain was used, and the sections were kept in methyl blue solution, and after 1 minute this methyl blue is also removed, that is remove the methyl green from the solution with the help of the blotting paper. Immediately after this step, 95% ethanol is allowed to sit on the gentle and is gently agitation for 1 minute, and removal of ethanol was again removed after 1 minute. Again 95% ethanol was added in the sections for 1 to 2 minutes and load them again. So we don't have to leave the sections for long in ethanol, as it may remove too much of the stain. The section should appear translucent to blue. After that, we have to transfer the sections in microscopic site with filter paper and blot the excess amount of ethanol from the sections.